So we know that cells, okay, have genes that drive cell growth, right? Yes? Yes? All right. So changes in those genes can cause uncontrolled cell growth. Yes? But changes in those genes can also cause apoptosis. What is apoptosis? Cell death. Okay? And the balance between the apoptosis and cell growth is what allows your cells to continue to grow and not go uncontrolled. Okay? But uncontrolled, that balance is controlled by what? Well, it's still going. It doesn't matter. It's not a right or wrong answer, so I don't care. Okay? But what is that balance is controlled by what then? Ultimately controlled by what? It's controlled by gene expression. Okay? If a cell produces too much of a gene, right, that drives cell growth, it's going to grow faster. If the cell produces there's an abnormality in a gene that helps control cell growth, there's going to be a problem with controlling cell growth and inducing apoptosis. Okay? So, cell growth by itself, right? What does this look like? The cell cycle. We went over this last semester, right? This is the normal progression of cellular growth. What parts of the cell cycle are there? All right, so we have G1. What else? S phase. Then G2. And what's this little one? Mitosis, or M phase. All right? And there's different stages of the cell cycle because cells are doing different things at that point in time. What's happening in the cell in G1? It's just growing and normally doing its job. But at some point, signals from the outside or internal signals, some type of stimulus, tells this cell to progress through G1 and enter into S phase. And S phase begins what? S phase, what's happening in S phase? Right? Replication of DNA. It's the synthesis phase of the cell cycle. All right? This point between G1 and S phase is very important. All right? This is called the restriction point. Okay? Once the cell progresses past G1 and into S phase, it's committed to replicate the DNA. All right? So it will go through S phase, and then there's another checkpoint at SG2, and either another checkpoint at G2M to make sure that everything's going, and then into G1. All right? That's the normal progression. Okay? These 2 points, these 2 parts where we talk about stimulus to grow, okay, and restriction points or ways to control growth is your balance which allows for normal cellular growth. All right? Yes, you need some cells to grow at a certain point to replace other cells, but you don't want them to grow uncontrollably, all right, or out of hand. And that balance is what allows normal cellular growth and prevents cancerous or abnormal cellular growth. All right? Questions on that? So, This is controlled by abnormal regulation of genes that affect the cell cycle. Okay? So the cell having its normal gene systems that help to provide cellular growth and drive cellular growth or control it normally can be altered. Those genes can be targets of mutations, problems, etc. And we're going to go through a couple types of those things that occur in cancer today. All right? But they're the same systems that are in embryonic development and in normal development in terms of progression of. All right? So, types of genes that are associated with cancer. All right. Years and years and years ago, when cancer was first discovered, and hundreds of years ago, first discovered and first was a topic of research. What the idea was, what causes this, and why are these cells different than other cells? And they looked at different sets of genes that drive cellular growth of these cells. All right, and they found a host of genes that are upregulated or they're turned on in cancer cells. All right, and these are genes that cause cancer. Okay, so any change in those genes or increased expression of those genes can cause uncontrolled cell growth and cause cancer. So they were termed oncogenes. Okay, oncogenes for the most part have a normal version of the gene in normal cells. Okay, so an oncogene is a abnormal version of a normal gene. All right, and so we talk about oncogenes as being cancer-associated genes and proto-oncogenes being the normal version of that gene in normal cells. All right, for instance, RAS. RAS is a growth factor. All right, and we're going to talk about RAS in the next couple slides. It's a growth factor, and it helps to drive cellular growth by phosphorylating cyclins and cyclin-dependent kinases that help to drive cellular growth and drive the cell cycle. All right? It is a normal gene that's produced by all your cells. Okay? There are abnormal forms of RAS. Some viruses, such as HPV virus, human papillomavirus, okay, which has an abnormal or a viral form of RAS. All right? The virus infects a cell, right, and transfers that viral form into the cell so that the cell continues to grow. Why is it important that the virus tells the cell to keep growing? What's that? So the virus can reproduce. So it's part of the virus itself and the way the cell is infected. But what does that do for the cell? Now it's producing a viral form of RAS. Is that more or less than it normally has? More. More RAS means faster growth, right? And as you know, HPV, that virus, is a common cause of what? Certain type of cancers. What type of cancer? How many of you, especially women, have had the HPV vaccine? All right, so you've got the vaccine, but you don't know what it's for? What it's going to protect you against? What? Actually, cervical cancer. All right? So as you get older, you're going to go to the gynecologist and get yearly exams, taking pap smears, looking at cervical cells to look for cervical cancer. Okay? That's what that's for. Okay? And the HPV vaccine helps prevent cervical cancer. Okay? So which gets right down to the idea of that viral RAS increasing expression of RAS in the cells and leading to abnormal cell growth. Okay? So RAS is an example of a proto-oncogene, okay? And the viral form of RAS is an example of a oncogene. All right? So one being normal cells, one being abnormal. All right? That is an example of an oncogene being sort of transferred from a virus to a normal cell. Now, oncogenes can occur through mutations of those proto-oncogenes, normal mutations that can be occurred. All right? So, oncogene arises from genetic change leading to either an increase in the amount or the activity of the protein product of the gene. Either way, using RAS as an example, if I increase the amount of RAS, what's going to happen? You're going to, so RAS normally signals and drives cell growth. If I have too much of it, it's going to drive the cell to grow faster, right? Yes or no? Yes. Okay, so now if I have more of it, or what if I increase its activity? It's going to drive the cell to grow, Cody, faster. Okay? Faster growth, all right, lead is not good, okay? So here are ways where a proto-oncogene can occur, okay? First, you have a normal proto-oncogene here, right? Here's normal proto-oncogenes in the cell, okay? Happy, doing their job. They're under their correct regulation. The proximal control elements in the DNA are doing their job. The distal control elements, transcription factors are there. Stimulus for the cell to grow normally. All right? But what happens if I take that gene, for whatever reason, a chromosomal abnormality occurs, and that gene moves to an area where it's regulated differently. All right? So that RAS, which is normally controlled in a certain manner, it now is moved to under control of a different promoter that is always on. Okay? There's an enzyme in your cells called glyceraldehyde dehydrogenase, or glyceraldehyde phosphate dehydrogenase, which is on in every one of your cells all the time because it's involved in glycolysis. Okay? So, glycolysis happens in all your cells. What if I take that RAS gene and I put it under the control of glyceraldehyde phosphate dehydrogenase? What's going to happen to that? Is the activity going to go up or down? It's going to go up, which is going to lead to abnormal cell growth. Okay? Now I have too much of that oncogene. Now it's an oncogene. Now I have too much of that RAS that's being produced. Leads to, all right, normal cell protein is now in excess. Too much of it. All right? Same thing can happen in terms of gene amplification. So somehow during mitosis and replicating chromosomes, maybe one of them gets an additional replication. Right? There are problems in mitosis that happen all the time. So that this gene, the RAS gene, gets amplified. Now instead of 2 copies, I have 4. What's going to happen to the amount of RAS in your cells? It's going to be inherently what? I have double the gene number of genes. How much of the protein am I going to have? Double, which means more protein to be active, more cell growth. Okay? So you can see how changing the proto-oncogene, the normal gene, into a abnormal gene so that there's too much of it leads to abnormal growth. All right? And then finally we have a proto-oncogene where there's a point mutation. What's a point mutation? Change in one spot, one amino acid, right? Or one base pair, one point, okay? 2 places that can occur. You could change the DNA in the control element. what's that going to do? What if that change in the DNA now makes it always on? So that the control, it never shuts off. Now what happens? Do I have too much or too little RAS? Too much. Leads to excessive growth. Okay? What happens if I do something within the gene itself that instead of now that changes how the gene is controlled, how the protein is controlled, so that it's always active and never shut off. The protein. Well, it's open.
Sorry.
That's okay. All right. So now instead of increasing the number of RAS molecules or proto-oncogene molecules that this part have done, instead, this point mutation has changed the activity. Now it's more active. Or you can't shut it off, or you can't break it down anymore. So it's always present. So in effect, it's a always on type of message. Right? The stimulus to grow is always there. Okay? These are the 3 or 4 ways where a proto-oncogene can be changed into an oncogene. All right? And we've been using RAS as an example. RAS is not the only one. Okay? There are hundreds of growth-related genes that can be changed into an oncogene. So proto-oncogenes to oncogene. All right? So we have movement to a position near an active promoter, may increase transcription, amplification, increase the number of copies. All right? Both of those lead to an excess, right? Too much of the growth factor, whatever it might be. And then point mutations, all right, can change its control, all right, or causing an increase in gene expression. Questions on that? So the cell sort of has a fail-safe, though. So that's one way of doing, right? We talked about the cell cycle and the fact that there's a point where the cell controls the gateway to abnormal or to cell growth, right? This is one stimulus for cell growth, but there's still a control mechanism, all right? And that control mechanism is sort of like the brake in your car. Think of the proto-oncogenes as being the gas pedal to make the car go, right? Drive cell growth, while the brake helps to slow it down, all right? And so the tumor suppressor genes, they encode proteins that help prevent uncontrolled cell growth, okay? And there's loads of these that are available too, all right? But they work to stop the cell from growing during those checkpoints, all right? They work to most likely repair damaged DNA. They control cell adhesion. They inhibit the cell cycle, all right? So they work to control cell growth. So now if a cell undergoes a mutation in a tumor suppressor gene that works to control cell growth and keep that sort of as a gateway, what's going to happen if that tumor suppressor gene no longer works? Abby? Right, so you're removing that control, cell growth is going to occur, right? Beyond, that stimulus is still going to be there to drive cell growth, but you're removing the brake, it's going to go uncontrollably now. All right? An example of a tumor suppressor gene. Have you guys heard of that? BRCA1. No? Okay. Breast cancer is one of the leading, most diagnosed cancers in the United States every year. Okay? And more than 50% of diagnosed breast cancers are associated with a gene mutation in this gene, in either BRCA1 or BRCA2. BRCA, what is that? It's breast cancer. It's a protein was first associated with breast cancer tumors. Okay? And they looked at breast cancer tumors as opposed to other tumors and found that there was a disruption in this gene that led to the formation of breast cancer. Okay? So more than 50% of breast cancers have a disruption in one of these two genes. Pretty important, right? BRCA1 is a tumor suppressor gene. Okay? And it works to control cell growth at inflection point. Okay? So you have two different ways. So I've been using the RAS gene. We're going to look at it again. All right? So we want to look at RAS again, okay, as a proto-oncogene, and then P53 as an example of a tumor suppressor gene. Right? And they are both common in human cancers. All right? P53 as a tumor suppressor has been found to have disruptions in greater than 75% of all cancers. All right? So no matter the type, likely so that P53 ends up becoming mutated at some point in time, whether you talk about colorectal, testicular, lung, breast, whatever it might be, whatever type of cancer we're looking at. P53 tends to be a target of that. So do you think P53 has an important role in normal cellular function? Yes, right? All right. So mutations in the RAS gene can lead to production of a hyperactive RAS, increased cell division. All right? So here we have sort of the two. So here's which one of these is the normal situation. Look at the diagram. Where's the normal situation? All right, so up top is the first one, right? So you have growth factor, some type of outside stimulus present in the blood, in the tissue, wherever it might be. Could be a hormone, whatever, right? Just signaling for your skin cells to divide, to produce more of cells for replace cells that have been lost, for example. All right, that growth factor is always going to be there. All right, because those cells should be growing at a normal rate. All right, RAS protein is activated. There's a couple protein kinases. There's what we call a signaling cascade, right, within the cell. Gets to the nucleus. Transcription factors are initiated. Genes are turned on that will produce or drive cell growth. All right, that's the normal progression. Now, growth factor is still there, but now I have an active or overactive or even too much RAS. Okay? RAS protein is overactive. It's going to induce that cell cycle or that cell signaling cascade, right? It's going to make this go faster. That's going to increase the transcription of these genes, and you're going to get overexpression of the protein. That overexpression of the protein is going to be important for cell growth. Cell growth ultimately is sped up or increased, right, beyond control. Okay? So that's a way of excessively driving cell growth. But we know that there's still the inherent or the cellular way of controlling this. Right? There's still the gateway there. So suppression of P53 or other tumor suppressors, right, can remove that gateway, and now the cell can off and run. Okay? So suppression of the cell cycle can be important for cause of damage to cell's DNA. So if a cell has damaged DNA, should it go through the cell cycle? Dean is saying no. No, right? And that's a checkpoint of the cell cycle to check to make sure replication is complete and DNA is complete, and what we call the fidelity of DNA, how well copied and how well structured the DNA is. All right? And part of the normal role of P53 is in this. All right? So P53 prevents the cell from passing on mutations due to DNA damage. All right. But when you get a disruption in that P53, more mutations are passed on. All right? And the cell, that sort of break is relieved, and cell cycle progression is uncontrolled. All right? So you prevent suppression of the cell cycle when P53 is mutated. Here's what it looks like. All right? Now, UV light is a normal exposure to DNA damage that we have every day. Right? Was down in Florida, exposed to wonderful sun. In Guatemala, probably exposed to some type of sun, right? So with exposure to sun comes what? Exposure to what? UV light. Okay? Your skin cells are exposed to UV light. That UV light causes what we call thymine dimers to form, which is a DNA damage point. Your cell recognizes this and works to create and repair mechanism to fix it or remove those dimers and make it right. Okay? So, your cells, if they can't repair it, they'll stop mitosis and prevents the proteins inhibit the cell cycle. That's the normal progression. But when you remove the control, that damage that's been incurred moves on to the next generation of cells. Who knows where that damage might be? Maybe it's in a proto-oncogene, right? If it's in a proto-oncogene and P53 is mutated, what do you get? Cancer. Okay? So there's a lot of control, sort of quality control steps that are put into the cell to prevent that from happening. Okay? What does this tell you about the cell in terms of control? Let me ask you the question in a different way. Is it likely that a mutation in one gene is going to cause cancer? Yes or no? Ben says no. Lauren says no. Anyone else? Sarah says no. How many of you say no? Okay. So most of you say yes. Right? Why? All right, why say no? Tell me. Kat, why no?
Has mutation in a single gene. It could be like a gene that doesn't do much and that will cause cancer, but that gene could have an effect on another. In like.
So another step down the road. All right. So what if I said? What if I made it a little bit more? What if I asked a mutation in one proto-oncogene does that cause cancer? Is it likely that a mutation in one proto-oncogene will cause cancer? Yes or no? Some people are yes. Yeah, you think so? Well, what's still there? All the regulatory factors that help control that. Right? Okay, so the cell has sort of checks for this. So there might not be one single mutation, but likely that it ends up being more than one. Okay? This is what gives the base of what's called the two-hit hypothesis. All right? That there has to be at least two distinct mutations that drive and cause cancer. Okay? One isn't necessarily enough. One can start the process, like Kat sort of alluded to. You have one, but then like P53. But now, because P53 is involved in repairing DNA damage, now from the subsequent daughter cells have a higher possibility of increasing their chances for developing mutations. More mutations in eventually proto-oncogenes, then you could lead to full-blown cancer. All right? So there's your multi-step model of cancer development. All right? Multiple somatic mutations are generally needed for full-fledged cancer. All right? And thus, incidence increases with age. As you get older, you acquire more mutations. Your cell's ability to repair DNA and recognize those mutations goes down. Therefore, you develop cancer as you get older. All right. This multi-step path is well supported, and we're going to use a colorectal cancer as an example to look at this. Okay. So colorectal cancer it may be seen as what's called a benign tumor. What's benign mean? Well, it's non-cancerous. It's abnormal cell growth, but it's non-invasive. It's non-metastatic. Tends to be, those cells are cancerous, right? Meaning that, well, I shouldn't say cancerous. It's a tumor, the cells are growing uncontrollably, right? Or at least increased cell growth, all right? But they haven't started to invade nearby tissues or metastasize to another, all right? In the case for fully cancerous cells, invasion, metastasis, right, abnormal cell growth, you're looking at on average, about a half a dozen mutations that have to occur in order for all that to happen. All right? But when you remove the ability of a cell to recognize and repair DNA damage, that chances of that happening increase over time. All right? These changes usually have one active oncogene and the mutation or loss of several tumor suppressor genes. All right? So the cell needs to have increased stimulus, right? The gas pedal gets stuck, and removing the gateway. Right? You're removing the brake. And that leads to full-fledged cancer. All right? So here's a model of colon cancer, the multi-step mode. All right? First, normal colon cells, right? They are happy. They grow in the epithelium, protect the underlying tissue. They are exposed to let's say, carcinogens all the time, from what we eat, what we take in, what we drink, okay? What we process through the body. All right, at some point in time, the first signal for this to occur, right, the first step is the loss of APC, anaphase promoting complex. What do you think anaphase promoting complex is involved in? Mitosis, right? So APC or another tumor suppressor is disrupted. All right? That leads to the ability of the cells to grow uncontrollably. All right? At least faster than nearby cells. All right? The cells that lost the APC or another tumor suppressor, they can grow faster than other nearby cells. That leads to a small polyp. All right? Now, because those tumor suppressors are normally involved in repairing DNA damage, that within that polyp that has lost APC, those cells have an increased chance of developing more mutations. Right? Maybe in one of those mutations, the RAS gene becomes activated. Now they grow even faster. Right? You get another loss of a tumor suppressor because now increased mutations. Right? So you end up with loss of tumor suppressor DCC. You have a larger benign growth that's now an adenoma. Right? Still hasn't developed the metastasis and invasion yet. Right? Loss of tumor suppressor P53 and additional mutations. Mutations that allow the cell to increase enzymes to break down the cells nearby the extracellular matrix. Right now it's invasive. It's become a malignancy. All right? And then there's a good chance that even that malignancy can be metastasizing to other areas of the body. All right, so this is a great model of showing you the disruption in multi-step, different processes along the line that lead to malignancy. All right, and the same thing can be said for breast cancer. All right, most breast cancers are they're usually characterized whether or not they're estrogen receptor positive, right, or negative, whether or not they have an EGF receptor or an EGF epidermal growth factor receptor problem, all right, and then also if they have a disruption in the breast cancer, the BCR-A, what we call BRCA1, BRCA2, or other tumor suppressors. Okay, all those mutations summatively, altogether, lead to full-blown cancer, invasion, metastasis, problems. All right, questions on that? And this is just, all right. So what does this mean for you? And especially in the form of BRCA1 and BRCA2. Okay? What happens if, and I'll give you the example of my sister-in-law and my nieces. Okay? My sister-in-law was diagnosed with breast cancer at 36. Okay? Full-blown aggressive breast cancer. She had the tumor removed, underwent aggressive chemotherapy, and luckily, she is a survivor. Okay? She has 2 daughters. All right? And typically with every cancer, they do a genetic analysis of what type of cancer it is. Right? Because that determines the course of action for therapy, especially in breast cancer. Okay? So if it's estrogen receptor positive, if it's BRCA2, BRCA1 negative, whatever it might be, because it determines whether or not you can use a hormonal therapy or a radiation or et cetera. Okay? It was found that her cancer has BRCA1 negative. All right? So here's mutations in the BRCA1 gene are found in at least half of inherited breast cancers. All right? And you can test these mutations. This is also found to be passed on. So now, what's the likelihood that my 2 nieces possibly develop breast cancer, ovarian cancer, reproductive cancers later in life? Pretty high, right? Higher than, I won't say pretty high. It's higher than the normal population of where their mother has a normal set of no mutations in the BRCA1 gene, right? If that mutation is passed on to both my nieces, right? So you may have what's called an inherited disposition, right, for developing cancer, right? So it increases your chances. So when you go into later on, you probably don't do it very often right now, but later on when you get older, you go in, you're going to go for a physical. Doctor's going to give you a history report. You're going to ask any history of heart disease, any history of diabetes, any history in your family of cancer. What type of cancer? Why do they do that? Why is that part of, I have all the PAs in here, okay? You're going to be asking these questions. Why are you asking these questions? Because you need to know, right? What's the predisposition? Is there a predisposition? Right? Is important. There's something that we should be looking for come years down the road when you have a sickness or something. Okay? Okay? And then finally, you can have DNA breakage can contribute to cancer, right? Ultraviolet radiation, cigarette smoke, they're all called carcinogens, viruses, you name it. Outside, this is a harmful world for ourselves. Right? Every day we are exposing our bodies to possible carcinogens. I see let's see, 1, 2, 3, 4, 5, 6, 7, 8, 10, 12 water bottles right now. What's the problem with water bottles? BPA. BPA is a known what? Carcinogen. Okay? So you have to make sure your water bottle now is BPA free. Don't let it sit in the sun. Don't put it in the microwave, blah, blah, blah. Because it is a known carcinogen, has been known to help cause cancer in laboratory animals. All right? Example of the type of exposure that we all have every day. All right? Questions on cancer and how it develops? No?